everyone. My name is Anastasia Lando, and I'm studying Chemistry 109, which is the chemical analysis of food. Today, I'll be presenting on the selection of techniques used in food analysis based on reading 1 and the statistical assessment of results of food analysis based on reading 2. Content. As I've mentioned in my introduction, that this presentation will be focusing only on the two readings. First reading is based on the selection of techniques used in food analysis, and reading 2 statistical assessment of results of food analysis. Introduction to food analysis. Food analysis is a scientific discipline that investigates what food is made of, how it behaves, and how it changes over time. When we talk about composition, we mean the nutrients and compounds present, like proteins, fats, sugars, and minerals. Structures refers to how these components are arranged, from visible cut pieces in cheese or to microscopic fat globules. Properties include flavor, texture, and stability, things that affect both enjoyment and shelf-life. This analysis is essential for research, such as developing a product, for the QCs or the quality controller, ensuring consistency and safety, and for product development where innovation meets consumer demand. Sample selection and preservation. Before any analysis can begin, we need a sample, but not just any sample. It must be representative, meaning it accurately reflects the whole batch of product. We can select samples in different ways. We can select samples through random samplings. This sampling it avoid bias. Systemic sampling might involve taking a sample every hour or from a specific location. Judgment-based sampling relies on the analyst's expertise. One selected sample must be preserved to prevent spoilage or chemical changes. That means we have to control temperature during refrigeration, moisture to prevent microbial growth, oxygen to avoid oxidation rather, and light, which can degrade sensitive compounds. Proper preservation ensures the sample remains stable until analysis. Extraction techniques in food analysis. Food is a complex mixture, so we often need to extract specific compounds before analyzing them. The goal is to isolate the analyte, the substance we are interested in, from the rest of the food matrix. Here are key extraction methods. First, it's digestion. Breaks down organic matter to release ions. Microwave digestion is fast. UV fertilizers uses radicals for cleaner breakdown. The membrane technique, like dialysis and ultrafiltration, separate compounds based on size or charge. The solvent extraction dissolves target compound. Supercritical fluid extraction uses CO2 under pressure, efficient and eco-friendly. Solvent methods use materials like silica to trap analytes, which are later eluted. Earth-based techniques capture volatile compounds from the air above a sample, ideal for aroma analysis. Each method is chosen based on the analyte's properties and the food complexity. Criteria for technique selection. Choosing the right analytical technique requires careful consideration. First, we assess analytical capability. Can the method handle the sample size? Is it cost effective? Will the sample be destroyed or remain intact? Next, we look at the performance metrics. Precision means consistent result. Sensitivity refers to detecting small amounts. Reproducibility ensures results can be repeated across labs. We also consider personal concerns, like is the method safe to use? Is it simple and fast enough for routine testing? Finally, we ask whether the method is official, validated by organizations like EOEC, or an in-house method which may be never but still reliable if properly validated. These criteria help ensure the technique is both scientifically sound and practically feasible. Categories of analytical techniques. Food analysis techniques fall into four major categories. Chromatographic methods, which includes GC, HPLC, and SFC. Spectroscopic methods includes UV, VIS, IR, NMR, and MS. Physical techniques and biological techniques. For chromatographic methods, separate compounds. Gas chromatography is great for volatile substances. HPLC handles non-volatile ones. Supercritical fluid chromatography, SFC, excels with lipids. Spectroscopic methods identify compounds based on how they interact with lights. UV-VIS measures the absorption. IR detects molecular vibration. NMR reveals molecular structure, and MS identifies compounds by mass. Physical techniques measure texture, structure, and thermal properties. Rheology studies flow. Microscopy reveals microstructure. Calorimetry measures energy content. Biological techniques use living systems. Enzyme sensors detect reactions. Immunoassays like ELISA identify specific proteins or lesions. Its category offers a unique insight, and often multiple techniques are often combined rather for comprehensive analysis. Chromatographic techniques. Chromatography separates compounds based on how they interact with stationary and mobile phase. GC is ideal for volatile, thermally stable compounds like lipids and pesticides. Polar compounds can be analyzed if chemically modified. GC by GC, comprehensive two-dimensional GC, enhances resolution by separating along two axes, useful for complex mixtures like flavors and fatty acids. HPLC is the most widely used ones. Widely used techniques for non-volatile compound. Reverse phase HPLC is especially versatile. Supercritical fluid chromatography, or SFC, uses CO2 under high pressure to extract lipids efficiently, through its less commonly used due to operational challenges. Spectroscopic techniques. Spectroscopy reveals how food molecules interact with light. UV-VIS spectroscopy measures how much light is absorbed at specific wavelengths, useful for compounds with chromophores. Fluorescence spectroscopy, it detects emitted light and is extremely sensitive, making it ideal for identifying trace contaminants like pesticides residues. IR, or infrared spectroscopy, especially the FTIR, is used on production lines to measure fat, protein, and moisture without sample prep. Techniques like ATR and DRIFT or drift enhance IR versatility. Raman spectroscopy complements IR by detecting changes in polarizability. It's particularly effective for proteins and nonpolar molecules. Advanced spectroscopy and mass spectrometry. Advanced techniques offer deeper insight into food composition, atomic absorptions, and atomic emission. Spectroscopy detects trace metals. ICP AES can analyze multiple elements simultaneously. Mass spectrometry identifies compounds by mass to charge ratio. This is often paired with separation techniques like your GC or HPLC. New ionization methods like ESI, EGN, APSI, and MALDI allow for analysis of complex molecules like proteins and antioxidants. NMR and ESR are non-destructive and can analyze internal structures. NMR is used for oil content and crystallization. ESR detects free radicals. Physical techniques in food analysis. Physical technique measure observable properties. Colorimetry quantifies food color based on hue, lightness, and saturation, important for self-life and sensory studies. Refractometry measures how light bends through a sample, useful for determining concentration in liquids like juices and sauces. Polarimetry analyzes how polarized light rotates through optical active substances, ideal for sugar and oils. Ultrasonic sensors. It measures how sound waves travel through food, revealing texture and composition. Through powerful ultrasonic imaging is time-consuming and routine. Electrophoresis and flavor analysis. Electrophoresis separates molecules based on charge and size. Gel electrophoresis is used for DNA and protein analysis. SDS page separates by molecular weight. Isoelectric focusing separates by charge. Capillary electrophoresis offers higher resolution in small volumes. It ideals for amino acids and vitamins. Flavor and odor are critical to consumer acceptance. GCMS identifies aroma compounds. GCO involves human sniffing during GC separation. APIMS measures volatile compounds during inhalation, offering real-time flavor release data. While sensory panels are still used, instrumental methods provide more objective and reproducible results. Rheology, structure, and thermal properties. Let's look at how food behave physically. Rheology is the studies of how food flows and deforms. It's important for texture, shelf-life, and processing. Food show both elastic and viscous behavior measured using tools like viscometers and shear tests. Structure is examined using microscopy. Light microscopy gives general views. SEM shows surface detail. DEM or TEM reveals internal structure, and CLSM creates 3D images with minimal prep. Thermal properties are measured using DSC, which tracks heat absorption during melting or decomposition. Bomb calorimetry measures total energy by burning the sample through manufacturers often estimate calories using ingredients data. Biological technique in food analysis. Biological technique use a living system to detect food components. Enzyme biosensors trigger measurable signals, optical, acoustic, or electrochemical when enzymes react with analytes. Microbial biosensors use generally modified microbes to detect substances like toxins or amino acids. Immunosensors rely on antibodies binding to specific antigens. The most common is ELISA, ELISA, which produces a color change that measured spectroscopically. These tools are used to detect contaminants, allergens, GMOs, and microbial threats. It's only critical for food safety and regulatory compliance. Introduction to statistical assessment. This is based on reading 2. Now we shift to the statistical side of food analysis. Every analytical technique, whether chromatography or spectroscopy, produces a measurement. We use statistics to relate that measurement to the actual concentration of a compound. There are two key needs. First, estimating the concentration in a single sample, often for comparison across labs or against standards. standards. Second, calibrating a new method using known standards to build a model that can estimate unknowns. This statistical layer ensures our results are both meaningful and reliable. Understanding uncertainty. Uncertainty tells us how confident we are in a result. For example, we might say we are 99% sure a compound's concentration is between 21.52 and 23.65 milligram per kg. Three main sources of uncertainty. Measurement error comes from steps like weighing or dilution, which introduces variability. Sampling error arises when food isn't uniform, like uneven additive distribution or seasonal variation. Calibration errors stems from flaws in the model used to interpret measurement. The broader the question, like estimating tannin in all commercial tastes, the greater the uncertainty. Determining and managing uncertainty. To manage uncertainty, we first define rather the scope of our study. Are we analyzing one sample or many? Are we using one analyst or several? Then we perform replicate analysis, repeating the test to see how results vary. To avoid bias, we use experimental design. For example, if 4 analysts use 2 instruments, each should both tools equally. Organizations like IUPAC, Eurochem, and NIST provide guidelines to ensure statistical rigor. This process helps us quantify uncertainty and improve confidence in our results. Replication and precision. To get a reliable data, we need replication. If we measure a compound 100 times, we'll get a solid estimate of its standard deviation, say 0.136 milligram per kg. But with only 5 replicates, that estimates could be way off. Each step in the analysis, like weighing, diluting, or measuring, introduces variability. By replicating each step, we can calculate its precision or how tightly the results clusters around the mean. This is the foundation for calculating overall uncertainty. Calculating uncertainty. Uncertainty is usually expressed as a percentage. For example, a flask might have 2% uncertainty, a balance 1%, and an extraction method is 5%. We combine this using statistical formulas to get overall uncertainty. I say 14.5%. Interestingly, small sources like 1% or 3% can often be neglected without affecting the total much. Manufacturers often provide specs like pipette range of 4.92 to 5.08 milliliters. We divide the range by 3 to estimate the standard deviation. This calculation only works if the method is consistent across samples, same equipment, same condition. Confidence and distribution. Confidence tells us how sure we are about result. In a normal distribution, 95% of values fall within 1.96, a standard deviation of the mean. So if our uncertainty is 3.2% and the measured concentration is 556.3 milligram per kg, we are 95% confident the true value lies between 52.77 and 59.83 milligram per kg. But with small sample size, we switch to the t-distribution, which adjusts for fewer degrees of freedom. For example, with 11 samples, we use 2.228 standard deviation instead of 1.96. Confidence intervals are essential for interpreting whether a compound exceeds a safety threshold or meets a regulatory limit. Reporting uncertainty. There are two main ways to report uncertainty. First, as a standard deviation, for example, saying a batch of flask has a volume uncertainty of 0.32 ml. More commonly, we report a range like 86.69 plus minus 3.27 milligram per kg. That plus minus 3.27 is twice the uncertainty known as the coverage factor of 2. This means we are about 95% confident that true value lies within the range. It's a simple but powerful way to communicate reliability. Accuracy and bias. Accuracy tells us how close our result is to the true value. For example, if the true concentration is 103.24 milligram per kg, and we measure 105.61 milligram per kg. Our errors is 2.37 milligram per kg. Precision, on the other hand, is about consistency. How tightly results clusters. A method can be precise but not accurate if there are bias, like a miscalibrated balance that consistently gives wrong values. Determining accuracy is harder than precision because we need a certified reference material or interlaboratory comparisons to know the true value. Comparing means and calibration. To test whether two procedures differ significantly, we compare the means using pooled standard deviation and calculate the statistics. If the t-value is high, the difference is likely real, not due to chance. For example, a t-statistics of 2.65 with 58 degrees of freedom gives a probability of 0.010, meaning we are 99% sure the procedures differ. Calibration is also key. It's how we relate a measured signal, like a chromatographic peak to a known concentration. In a classical calibration, we use a series of standards to build a model, then apply it to unknowns. These ensures our measurements are not just precise but are meaningful. Classical versus inverse calibration. Calibration links a measurable signal like absorbance of peak area to the actual concentration rather of a compound. In a classical calibration, we assume all errors is in the response variable x and the concentration C is known precisely. In inverse calibration, we assume the error is in the concentration. Often more realistic when standards are prepared in-house, it's good practice to build both models and compare predictions. If they diverge, it may indicate outliers, samples that were prepared incorrectly or measured under poor conditions. This should be investigated or removed. Error and analysis in calibration. To assess calibration quality, we analyze residuals, the difference between the predicted and observed values. A residual plot helps spot trends like increasing error with concentration or outliers with large deviation. We calculate the root mean square error to quantify how well the model fits the data. If the relationship isn't linear due to the detector overload or signal noise, we may need to dilute samples or adjust instrument settings. This ensures our calibration is valid across the concentration range. A replicate error versus model fit. To validate a calibration model, we compare replicate error variation within the repeated measurement to the lack of fit to the model. With only a few replicates, we get a rough idea of experimental uncertainty. For regulatory purposes, we need many more replicates to ensure accuracy. We calculate average response at its concentration level and use this to compute total replicate error and residual error. This helps determine whether the model is truly capturing the relationship between concentration and response. Lack of fit and model validation. To assess how well our calibration model fits the data, we calculate the lack of fit error by subtracting the replicate error from the total residual error. We then divide each error by its degree of freedom to get average errors. The F ratio compares these averages. If the F ratio is less than 1, the model fits well. In our example, the F ratio is 0.478, meaning the lack of fit is smaller than the replicate error, so the model is valid. If the lack of fit were larger, we'd use an F-test to determine whether deviation is statistically significant. Confidence interval in calibration help us understand confidence intervals help us understand how reliable our predictions across the concentration range. Confidence is highest near the center of the calibration range, say 10 mole per liter in a 5 to 5 mole per liter range. Prediction outside this range have wider intervals. For example, a predicted value of 18 mole per liter might have 95% confidence range of 14 to 22 mole per liter. We use the t-distribution to calculate these bounds, adjust for degrees freedom. Graphics plots of the calibration line with confidence bands help us to spot outliers and nonlinear trends. This approach can also be applied to unknown samples, giving us a quantified uncertainty for its prediction. Conclusion. To wrap up, food analysis is a multidisciplinary science that combines analytical techniques with a statistical validation. Choosing the right method depends on the compound food metrics and the goal of analysis. Uncertainty, accuracy, and calibration are key to ensuring results are both meaningful and reproducible. Confidence intervals and error analysis gives us the tools to interpret and trust our data. Thank you.